as we are now seeing right now, even with the hantavirus outbreak, we don't have those facilities everywhere, right? And Ebola is one of those types of diseases that is not just a typical room in a hospital that you would quarantine somebody. So Angie, since you have been in high security BSL containment labs, you understand that Ebola requires very specific attributes of a proper place to isolate and quarantine. Can you describe that?
Yeah, I mean, so the difference between working in a containment lab and a ETU or working in a patient facility is actually quite different. So I can't really comment on the patient facility aspect of it, but I can certainly comment on the fact that this is really to mitigate the same risk. And that is that even though Ebola is never going to be transmitted from person to person, as I said, it can actually be transmitted to a person via the aerosol route. So the first cells that Ebola virus infects in the body are called macrophages, and there are a lot that are resident in your lungs. They leave your lungs after they get infected, so that's why they're not still there shedding virus into your airway, but you can get infected that way. And in fact, I've worked on experiments where we experimentally infected non-human primates, and we could get uniform lethality in adult non-human primates with as little as one plaque forming unit. So that's one infectious unit of Ebola virus.